So we'll open up this session for questions. Dr. Ampel.
Well, thanks. All really good talks. John, this is to you. Excellent review. As we discussed, this is more of a discussion point. We've sort of been hoisted on our own petard and Coxie. As you've indicated, we don't have any drugs that we can current drugs that we can absolutely say work. We can't do inferiority studies. And we currently get our drugs from other studies for other fungi. But that's very problematic because that requires most of us clinicians to go for prior authorization, and often those are disapproved or the patient has to pay large amounts. So I'm just going to make a pitch, however difficult it is. I really think we need to try to have studies that allow us to get these new agents FDA approved for coccidioidomycosis. Because otherwise we're just always left behind the 8 ball with costs and prior approval, and then we never get any really good studies. We're always just doing retrospective studies which have all those problems. So just a comment regarding that.
Yeah, I'd like to actually maybe add to what Neil just said. You know, as I was preparing this talk to discuss this, and as John Rex sort of also found, not just for this talk, but as he's been trying to develop his drug for F2G, it's really hard to meet the bar that the FDA has set as a routine matter for how drugs are going to be declared efficacious. And I'm actually not trying to be cynical about the idea of approving the drug for some other indication and then doing phase 2 trials with large N's, or 50 to 100 sample size. And if you have 2 of those, you have a chance to see error bars around response rates. And I understand there's no comparator, although you have historical comparisons. But, you know, in fact, I think there could be an interesting dialogue with the FDA about what constitutes necessary evidence for efficacy for this disease. That we've been able to manage our patients with just that kind of data. It would be, I think, valuable if the FDA had some way to have the labeling reflect that. Because you can do whatever the labeling says, and they wouldn't have to say you've proven differences between this or that, but something else. I think some creative policy management here would be really fruitful. And part of the problem, and I'll stop, but part of the problem is the drugs we've used up to this point have all been static. None of them cure this disease. And the whole question about treating early Coxie, if we had a drug known to be curative, the whole paradigm would be flipped. We would be wanting not to wait till we saw the complications. We'd want to identify early infections, get rid of it, and be done with the disease. And so I think if we get newer drugs that are cidal, this whole question of who to treat would get revisited.
I see John Rex there too. I don't know whether he had a question before me or not, but I'd make the point that isobuconazole was approved largely for Aspergillus, excellent trial. And some people here were clearly involved in that heavily. We got an approval for mucormycosis with isobuconazole. And as far as I know, there were 34 patients that got that approval. So the FDA's brain can be bent. The only question is, are there any Coxie people that can bend it?
John, John Rex, do you have any comments on the isobuconazole approval? We're using the historical mucormycosis data set.
I mean, the ISO approval is it's a relatively well-trodden pathway. they were able to do a standard randomized control trial in invasive aspergillosis with a non-inferiority margin that's pretty tight. It's a big study, so I think it was about 600 subjects, and it only took 5 years to run. And then adding on a second fungus based on, but really it leverages the primary data. It leverages the fact that there's an IARCT. And in many ways, that's similar to what happened with caspofungin 20 years ago, where there was, you may not remember it, but there was a study of esophageal candidiasis versus amphotericin, believe it or not. And that was a nice randomized study that clearly demonstrated that caspofungin worked. And then the aspergillosis component was an open label data series that, the diagnostics at the time were not very good, but it was the best you could do. And there was a novel class, there was a need to carry that forward. And so it's a clear-cut pathway, and the FDA's rules have not changed. The core element is, and you just have to tattoo this on the inside of your eyeglasses, substantial evidence of efficacy based on adequate and well-controlled clinical investigations. But don't ever call that a regulatory hurdle, because it's the same thing all of us want too. But the trick is, how do you calibrate? And I think that's what Royce is kind of getting at there. How do you calibrate? Where do you set your p-value? How strong does it have to be? And that's where, I spent a lot of time yesterday talking about LPAT, why I think that's a tool to study, because the principle there is when you're in a tough corner, you can focus on the risk benefit in a smaller defined population, and the risk benefit in that population can be more sharply etched because you've got a really hard to treat disease. So I think that's the kind of thing to think about. That's certainly what I'm thinking about.
I'll ask a question to John. So I don't know the data too well, but the LPAT approvals seem to me, the two that we have in infectious disease, seem less contingent upon a large data set for safety. So the Arikayce and the TB drug, I don't know, had a big trial to go with them. So they really were just more limited, right? So I think the FDA can be kind of persuaded with need.
Right. So the one that I can most readily quote is the TB drug. It's 100 patients, but they were treated for reasonably long periods of time. And I don't really remember the safety signal for that compound. Whatever it was manageable. And the context was absolutely, totally untreatable TB, which never, ever, ever gets better on its own. And they went into that study with a predefined hypothesis about a particular 3-drug combination. It was 2 approved drugs plus the third that had done spectacular things in animal models before doing this. So there was a recognition that this 3-drug combination might really do something, and it did. And so, that's the whole thing about LPAT is you wouldn't normally describe 100 patients worth of data as a big enough safety database, but it's not like there are gazillions of these XDR-TB patients, thankfully. And for them, for that limited and well-defined population, the risk-benefit of that drug is appropriate. Not appropriate for a larger population, a different population, not for every Tom, Dick, and Harry, but for that group, yeah. And that's the beauty of the LPAT language. In a sense, the FDA has always had, they intrinsically have that flexibility, but the LPAT language has made it more sharply etched. Here's a way to talk about it. Also a way to label it. If you look at those 2 compounds, you see the words limited population in big type sort of all over the label to remind you that this isn't for every Tom, Dick, and Harry.
I'd like to make a quick comment to add to what John was saying. I showed you those enrollments to the MSG, the NIH MSG studies, and they were quick because those drugs were not yet on the market. And actually, the randomized trial of flu versus Itra was when they were on the market, and we had to call the study at 198 instead of 200 because getting those last study patients on study, the drugs were so available we could not enroll them. So the strategy really should be at the beginning when the drug is not yet on the market. If you really want to get, the collaborative groups that enrolled to COXI trials have no trouble. These are patients that come to specialists, and the specialists enroll the patients. But only if the drugs can't be obtained off-label already in the pharmacy. And so I would be hoping that companies that are trying to develop it, if they were trying to get something other than a randomized controlled trial kind of approval, that they do it early in the approval process rather than say, I'll do it after we get the drug on for whatever other disease they're working on.
Thanks, John. So we have one question online that comments, are the sertraline MICs you mentioned equivalent to what one might experience with typical antidepressant dosing, which is 50 to 100 milligrams daily? I'll just answer that one if that's okay. We get asked that question a lot, and so I've looked this up before. In the serum drug concentrations you get with standard sertraline, which is Zoloft, are about 1 to 2. So to get levels of this high, you need to use about 2 to 3 times the normal Zoloft dose for patients, which they don't tolerate well. So, and we also have the Cryptococcus data, which has been published in Lancet, and sertraline didn't add a whole lot in that study. So I'm not sure that repurposed sertraline is going to answer a lot of our burning questions in COXI. So, yeah.
So George Rutherford, University of California, San Francisco. I have a question for Marcos. My impression was that in the Sertão, the part of Brazil we're talking about in POE, that a lot of that disease was in HIV-infected patients. At least historically, it had been. And with the improved antiretroviral therapy, are you seeing a declining incidence in that part of the country?
So you mentioned that. So I did mention that we didn't see any HIV co-infection with COXI in our study group.
Huh.
Yeah.
The national government's reported it consistently for a long time as a big OI in that part of the country. So that's good.
Yeah, but like for for COXI, we don't see. But actually, for histoplasma, it's a big problem, right? So if you look for CRI state, so CRI state has one of the highest mortality rates of histoplasmosis associated with HIV people. And the main problem there is the delayed diagnostics. So we do see increasing of antiretroviral therapy, of course, some more political efforts going towards that. But actually, we do see if we do have a delayed diagnostics, the mortality rates of, for example, histoplasmosis is increasing there. So that's and I think that it's great that we actually has a good antigen testing for those particular cases from IMI Diagnostics, for example, that we are now able to detect histoplasmosis cases earlier, and then we can save more lives. That's my point.
Marcus, I'll ask you a question we can probably all agree on. How many species of Posidoceae do you think there are?
Oh my gosh, this is a very good question. I mean, the point is we do observe some genetic isolation. We do see some population structure within Posidoceae. What we can actually do, like to define if it's a novel species or not, like giving names to the actors, right? We need to do more testing, and for now, I don't know if it's a good time for actually changing taxonomy within Posidoceae. For example, we do know that the population from Arizona, they might have some clinical differences between Arizona and Texas and Mexico patients, but at least we want to first better understand about how much consistency is there about these isolated populations to actually try to give more species. And there is another testing that we can do. It's actually calculating not only the pi values, which is the average nucleotide diversity, but actually to calculate nucleotide diversity between those clades. If we have if the pie it's greater than the difference between those populations, maybe we can start like giving novel names and define novel species. But the first step, it's actually like getting a better picture, a greater picture about what's going on within Posadaceae to in the future maybe talking about novel species. But now we're investigating genetic diversity, and that's all we can do.
Well,
That's not complicated
Coccidioides
things right now.
caribbeensis just sounds so friendly and.
Oh, maybe. Right?
Well, we will wrap this up. Everyone's ready for lunch. I just want to thank again all of our speakers for their time and excellent presentations today. And we will see everyone back here shortly.